So we are talking about signaling systems that are hot-wired between the cell surface and the nucleus. We're going to look at another example of that. Transforming growth factor beta family, receptor family. So transforming growth factor betas, they consist of a large number of structurally related secreted dimeric proteins, and they play an important role during development. They regulate pattern formation, influence various cell behaviors, for example, proliferation, differentiation, extracellular matrix production, and cell death. We have talked about apoptosis. In adults, these signaling molecules can initiate tissue repair and also play a role in immune regulation. They act through enzyme-linked receptors that are single-pass membrane proteins. Let's look at that. Basically, 4 of these receptors have to come together to form a complex when they're activated. They are type 1 receptors and type 2 receptors. TGF-beta binds, first of all, type 2 receptors, type 2 TGF-beta receptors, 2 of them, and when this binding takes place between 2 type 2 TGF-beta receptors, they recruit and phosphorylate type 1 receptors. You can see here in the image, we are only showing one of these type 2 receptors. There are 2 of them, and there are 2 of type 1 receptors. For the sake of simplicity, we are showing only one type 2 and one type 1 TGF-beta receptors. So they make type 1 receptors. When they're phosphorylated, they can bind a special protein in the cytoplasm, and it phosphorylates that protein. The name of that protein is SMAD. Now SMAD, there's not one type of SMAD. There are several types of SMADs. For example, TGF-beta receptors phosphorylate SMAD 2 or 3, whereas BMP receptors, BMP stands for bone morphogenic protein, categorized in TGF-beta receptor family. So BMP receptors, bone morphogenic protein receptors, phosphorylate SMAD 1, 5, and 8. So SMAD 2 and 3 are phosphorylated by TGF-beta receptors, whereas 1, 5, and 8 are phosphorylated by the BMP receptors. One of these SMADs then dissociates from the receptor and binds to SMAD 4. So here we say SMAD 2 or 3 if this was TGF-beta. If it were BMP receptor, it could be SMAD 1, 5, or 8. So anyways, after our SMAD binds SMAD 4, it could be 2 or 3 in case of TGF-beta receptor. This complex then moves to the nucleus, and here it can recruit other gene regulatory proteins and activate transcription of specific genes. So again, here we have a receptor complex forming at the surface of the cell and sending a molecule straight to the nucleus to influence the transcription. So there are shortcuts to signaling from the plasma membrane all the way to nucleus, and we have seen 2 examples of that.